Acid-base balance. The maintenance of a stable hydrogen ion concentration in body fluids is essential to life.
What is an acid? An acid is a substance that donates a cation or accepts an anion or electron. Meanwhile, a base is a substance that donates an anion or accepts a cation. Enzymatic activity and protein structure are frequently sensitive to pH. In any given cellular compartment, pH is maintained to allow for maximal enzyme or protein efficiency. Look at this illustration.
Effects of low pH: 1. Hyperkalemia due to loss of potassium from cells in opposed to renal loss of potassium. 2. Loss of smooth muscle tone and hence BP drop. 3. Loss of mineral from bone leading to osteoporosis. When discussing effects of high pH, tetany is a significant feature. When H+ is reduced, calcium binding to albumin increases and hence free calcium decreases. Ultimately, firing threshold of nerve axon moves closer to resting membrane potential, which is easier to excite.
Control of acid-base balance is via 3 mechanisms. 1. Buffering, 2. Respiratory regulation, 3. Renal regulation.
Respiratory acids are mainly due to carbon dioxide production leading to bicarbonate acid production. Metabolic acids. Non-volatile acids the body produces are known as fixed acids. These acids are usually referred to by their anion. All acids other than hydrogen carbonate are fixed acids. A buffer is a substance which has the ability to minimize changes in pH by reversibly binding to H+. A buffer will exist in 2 forms. The body has a huge buffering capacity. This system is immediate in effect. Now we'll look at major buffer systems in the body. The major buffer system in interstitial fluid is bicarbonate. Bicarbonate and hemoglobin are the main buffers of blood. Unlike in blood and ECF, proteins and phosphate are the main buffers within cells. In urine, phosphate is responsible for most of titratable acidity, while ammonia is important for formation of ammonium ions.
The major buffer system in ECF is the bicarbonate buffer system. This is responsible for about 80% of extracellular buffering. It is the most important ECF buffer for metabolic acids, but it cannot buffer respiratory acid-base disorders. Illustrated version of bicarbonate buffer is as. When substituted to Henderson-Hasselbalch equation, it is as this. The bicarbonate buffer system is an effective buffer system despite having a low pKa of 6.1. Look at the chart below. Phosphates are important buffers intracellularly and in urine where their concentration is higher. Phosphoric acid is triprotic weak acid and has a pKa value for each of the 3 dissociations. Protein buffers in blood are hemoglobin and plasma proteins. Their buffering is by the imidazole group of the histidine residues, which has a pKa of 6.8. This is suitable for effective buffering at physiological pH. Hemoglobin is about 6 times more important than the plasma proteins as it is present in about twice the concentration, contains about 3 times the number of histidine residues per molecule. All buffer systems which participate in defense of acid-base changes are in equilibrium with each other. This is known as the isohydric principle. At tissue level, deoxygenation of the blood increases its ability to carry carbon dioxide. This property is the Haldane effect. Conversely, oxygenated blood has a reduced capacity for carbon dioxide. For this reason, deoxyhemoglobin is a better buffer than oxyhemoglobin. Respiratory regulation of acid-base balance.
Respiratory regulation refers to changes in pH due to PCO2. Changes are from alterations in ventilation. Carbon dioxide is lipid-soluble and crosses cell membranes rapidly, so changes in PCO2 result in rapid changes in H in all body fluid compartments. Arterial partial pressure of carbon dioxide is proportional to carbon dioxide production by the body divided by alveolar ventilation. This effect is a reflex mechanism. Here, the stimulus is arterial PCO2, sensed by central and peripheral chemoreceptors. The central integrator is the respiratory center in medulla, while effectors are the respiratory muscles. Renal regulation of acid-base balance. In acid-base balance, the kidney is responsible for two major activities. 1. Reabsorption of filtered bicarbonate. 2. Excretion of the fixed acids, acid anion, and associated H. This is done via two major mechanisms. 1. Proximal tubular mechanisms. 2. Distal tubular mechanisms. The proximal tubular mechanism is responsible for 1. Reabsorption of the bicarbonate filtered at the glomerulus. 2. Production of ammonium. Daily filtered bicarbonate equals the product of the daily glomerular filtration rate and the plasma bicarbonate concentration. This is between 4,000 to 5,000 millimoles per day. About 85 to 90% of bicarbonate is reabsorbed in the proximal tubule. The rest is reabsorbed by the intercalated cells of the distal tubule and collecting ducts. This is an illustration of bicarbonate reabsorption along the nephron. H+ and bicarbonates are formed from CO2 and H2O in a reaction catalyzed by carbonic anhydrase. The H+ leaves the proximal tubule cell and enters the PCT lumen by two mechanisms. 1 via ANA plus H+ antiporter and 2 via H+ ATPase, proton pump. Under the influence of aldosterone. This is an illustration of Na plus H+ antiporter and H+ ATPase proton pump. Filtered HCO3 cannot cross the apical membrane of the PCT cell. Instead, it combines with the secreted H under the influence of brush border carbonic anhydrase to produce carbon dioxide and H2O. The carbon dioxide is lipid-soluble and easily crosses into the cytoplasm of the PCT cell. In the cell, it combines with OH to produce bicarbonate. The bicarbonate crosses the basolateral membrane via a sodium bicarbonate symporter. This symporter is electrogenic as it transfers 3 bicarbonate for every sodium ion. In comparison, the sodium H antiporter in the apical membrane is not electrogenic because an equal amount of charge is transferred in both directions. The basolateral membrane also has an active sodium-potassium ATPase pump. The net effect is the reabsorption of 1 molecule of bicarbonate and 1 molecule of sodium from the tubular lumen into the bloodstream for each molecule of H secreted. Look at this illustration. Very important slide, discussing AMCQ. The major factors which control bicarbonate reabsorption are 1. Luminal bicarbonate concentration, 2. Luminal flow rate, 3. Arterial PCO2, and angiotensin II, via decreasing CAMP. An increase in any of these 4 factors causes an increase in bicarbonate reabsorption. Meanwhile, parathyroid hormone increases CAMP and decreases bicarbonate reabsorption. Ammonium is produced predominantly within the proximal tubular cells. Ammonium is produced from glutamine by the action of the enzyme glutaminase. The processes involved in distal tubular mechanisms are formation of titratable acidity, addition of ammonium, NH4, to luminal fluid, and reabsorption of remaining bicarbonate. Titratable acidity represents the H which is buffered by phosphate, creatinine, ketoacids. Transport processes in the thick ascending limb are 1. Apical sodium bicarbonate antiport. 2. Basolateral sodium bicarbonate symport. 3. Sodium potassium ATPase pump. Moreover, the cells in this part of the tubule has carbonic anhydrous. There are some key differences between transfer between PCT and DCT. The net acid excretion in the urine is equal to sum of the TA, and the difference between ammonium and bicarbonate concentration. As pH falls, the three factors involved in increased H excretion are increased titratable acidity, increased buffering by phosphate and other organic acids, increased ammonium excretion. Bicarbonate reabsorption is complete, so none is lost in the urine.